a low-cost cell migration assay based on gelatin for studying the dependence of ECM stiffnesses. For example, breast cancer is the most commonly diagnosed cancer affecting women worldwide and the second leading cause of cancer death. Each year there are 2 million newly diagnosed breast cancers. The cancer prognosis does not only depend on the subtype, but also on the microenvironment of the cancer cells. An important component of the microenvironment is the extracellular matrix. In the human tissue, the extracellular matrix is a very complex network of biomolecules which encloses the cells. The ECM is important for many cell functions, for instance forming tissue and organs, control of cell proliferation, cell differentiation, possibility for protein adhesion, influence on tissue reformation and storage for regulating proteins. Depending on the composition, the matrix varies in stiffnesses. Emergent studies suggested that the stiffness of the ECM is a key player for increased cell movement, which leads to a high risk of developing metastasis and decreases the chance of survival. We hypothesized that the migration rate of cancer cells is lower in a softer ECM model than in a stiffer one. Unfortunately, only very expensive and complex ECM models are available. Therefore, our purpose was to investigate in the development of a low-cost and convenient gelatin-based ECM model. The mechanical and chemical properties of gelatin can be modified using various kinds of crosslinking agents. We used GTA as the crosslinker. This enabled that gelatin is stable at 40 degrees and to generate 3 different stiffnesses. GTA is very toxic for cells, therefore it has to be removed completely with washing in PBS. Between the washing steps, the GTA concentration was quantified with schiffsche reagent for aldehydes by photometric measurements. The results showed that the 4% gelatin 1.5% GTA gel is the softest and the 5% gelatin 0.3% GTA gel is the stiffest. After no GTA was detectable, gels were used for cell culturing. Therefore, 3T3 fibroblasts were used. In comparison, cells attach on the 5% gelatin 0.3% GTA better than on the other two gels. Cells on the stiffer gels don't spread out very well and stay in spherical shape. The morphology of fibroblasts showed significant change. Therefore, it indicates that the stiffness influences the cell attachment.